The topic of this video is Claudin-1 dependent destabilization of the blood-brain barrier in chronic stroke. To begin, this paper explores the effect that blood-brain barrier permeability has on stroke recovery and how the permeability is modulated by the Claudin family of proteins. Previous research has shown that damage to the blood-brain barrier following a stroke has been associated with increased paracellular permeability. As some background, a stroke occurs when the blood flow to the brain is decreased or stopped. This causes the brain to lose its supply of oxygen and nutrients, leading to cell death. There are two types of strokes that can occur: ischemic strokes and hemorrhagic strokes. The first type, ischemic, happens when blood vessels in the brain are clogged or narrowed, leading to reduced blood flow. This can happen if fatty deposits build up or debris that has been traveling in the blood gets lodged in blood vessels. The second type, hemorrhagic strokes, occur when a blood vessel in your brain leaks or ruptures. The next important background information is on the blood-brain barrier. The blood-brain barrier is a semi-permeable border that occurs in the brain. Its role is to separate blood from the brain and the cerebrospinal fluid in the central nervous system. It keeps out harmful toxins and pathogens from entering, while still allowing necessary nutrients and hormones to enter the brain. Following an injury or over the course of a disease, the barrier may become leaky, making it less effective at keeping these harmful substances out. A way that the blood-brain barrier adjusts its permeability has to do with Claudin proteins. Claudin proteins are structural proteins that occur in the tight junctions of the blood-brain barrier. They help to prevent paracellular diffusion of small molecules and have been seen to decrease the blood-brain permeability following their expression post-stroke. So knowing the effect of these proteins on permeability in the blood-brain barrier, the researchers hypothesized that an increase in these Claudin proteins would help the blood-brain barrier recover faster post-stroke. The first experiment looked at the effect of inducing a stroke in mice on brain function and the permeability of the blood-brain barrier. A transient middle cerebral artery occlusion was used to induce focal cerebral ischemia. They next tested neurological deficits using a modified neurological severity score from 1 to 14. Ipsilateral turning was then measured by allowing mice to run into a wall. The infarct size was then measured by killing the mice and using crystal violet to view volume size, with contralateral hemisphere volume minus ipsilateral hemisphere volume measured injury volume. The first test was an infarct volume test done via Nissl and fluoride B staining. It was found that the infarct size was decreasing over time. The next test was a neurological deficits test, where the mice were seen to improve over the weeks but never recovered to their original baseline. Following that, they did an ipsilateral turning test where the sham control test remained steady around 50%, while the stroke group about 90% of the time would do ipsilateral turning. The next test they did was on transfer coefficients for insulin and dextran. It was seen that insulin and dextran were expressed mostly in the ipsilateral region, with dextran falling off dramatically during the third week. The last test they did was to look at the amount of paracellular fibrogen inside the mice. It was found that the fibrin density post-stroke compared to the sham control rose dramatically. The second experiment looked at Claudin-1 and 5 expression and how it was correlated to the transfer coefficient of insulin. An RT-PCR analysis was used to look at the expression of Claudin-1 and Claudin-5 mRNA following stroke. A Western blot and semi-quantitative dystrophic analysis was then used to look at the expression of Claudin-1 and Claudin-5 normalized to GAPDH. Correlation between the expression of Claudin-1 and 5 was then compared to Ki of insulin. Immunostaining was then done to look at Claudin-1 and Claudin-5 in relation to fibrogen. This was then correlated to the density of the fibrogen in the extracellular area. The first test shows that Claudin-1 mRNA was only expressed in the ipsilateral region and started off high but tapered off over time. When looking at Claudin-5, it was found that mRNA started out low but increased over time, but was found in both regions. It was even found in the sham control. When the proteins were looked at and normalized to GADPH, similar results were found. However, the protein in Claudin 5 stayed the same over time and never increased. When looking at Claudin 1 and insulin, comparing them found a positive correlation, whereas one increased, the other would increase. When Claudin 5 and the transfer coefficient of insulin were compared, however, a negative relationship was found, whereas when Claudin 5 expression increased, the insulin transfer coefficient actually decreased. When looking at the comparison between the amount of extravascular fibrogen in Claudin 1 and 5, it was found that Claudin 1 had a positive relationship with the amount of fibrogen, and Claudin 5 had a negative relationship. This experiment looked at the effect of inhibiting Claudin 1 with peptide C1C2. Again, mice following reperfusion were treated with C1C2, and the permeability coefficient of insulin was measured. Next, supersolution images were taken in microvascular endothelial cells following ischemia and perfusion using C1C2. Next, immunostaining was done using various Claudin 5 elixifluors. Another turning test was then administered to see the effect of C1C2 against the control peptide group. Next, a protein array test was done to see the effects of C1C2 on pro-inflammatory mediators in microvascular endothelial cells. And lastly, the transfer coefficient of insulin was tested following stroke when treated with C1C2. The first graph marked A shows Claudin 1 inhibition on the permeability coefficient of insulin. It was seen that when the peptide C1C2 is used, there is a marked drastic decrease in insulin permeability coefficient when compared to when Claudin is overexpressed. This was tested on ischemia and reperfusion injuries over a 48-hour time period. The effect was also seen there when C1C2 is used. The insulin permeability coefficient is about half with the peptide than without. This stays true at the 24 and the 48-hour period. Supersolution images marked C show that when C1C2 is used, this displaces Claudin and allows Claudin 5 to be restored. The picture showing immunostaining showed that the use of C1C2 reduced the co-localization of Claudin 1 with Zondel 1. However, this allowed Claudin 5 to increase its co-localization with Zondel 1. The bar graph to the right, marked E, shows a protein array from various proteins across the study and the effect C1C2 had on them. It was seen that the depletion of Claudin 1 caused the expression of pro-inflammatory cytokines to go down. The next study shows the effects that C1C2 had on the transfer coefficient of insulin. It was seen that the transfer coefficient of insulin was greatly decreased by the use of C1C2. The last test they used was an ipsilateral turning test with C1C2. It was seen that when C1C2 was used, although initially they showed more ipsilateral turning, by the end of the 4 weeks, they were basically at baseline with the sham control. It was found that a stroke increases the permeability of the blood-brain barrier and leads to neurological deficits. This could be seen in the tight junctions of the blood-brain barrier, where many of its proteins had been depleted. One of the reasons for the increased permeability of the blood-brain barrier was found to be the expression of Claudin 1, a protein found in the tight junctions. When this protein was upregulated, it was found to be associated with higher transfer coefficients and higher permeability coefficients of various molecules. An issue with Claudin 1 is that it causes a decrease in the expression of Claudin 5, a protein that was found to decrease the permeability coefficient and transfer coefficients of various molecules. The expression of Claudin 1 also had the effect of causing Claudin 5 to not be able to localize with the Zondel region, causing more leakiness. This makes Claudin 1 an important target for therapeutic therapies. So, it was found that the use of inhibitor C1C2 peptide was able to decrease the expression of Claudin 1, which caused the upregulation of Claudin 5. This is an important target because today most therapies only focus on fixing the cause of clot itself and not taking care of clot recovery. So, in the future, if a peptide could be used to help decrease the expression of Claudin 1, it would be beneficial to stroke recovery. This could be seen in new clinical trials. This page shows the citations I used in my video project.